Okay. So this is an overview, or we can say review of the nephron. Okay. So we've got our arterioles here. Afferent arteriole. Efferent arteriole. Glomerulus. And then our Bowman's capsule. Right, then we've got the proximal convoluted tubule or the proximal tubule. The loop of Henle. The descending limb, the ascending limb. The distal tubule, and the collecting duct. Okay. Okay. So let's go through each of these areas here and talk about the important parts to bear in mind here. So the first part of this is the glomerulus. All right, so we've got the glomerulus, we've got the Bowman's capsule, and let's talk about the process that happens here. So we know that the process here, first of all, this is located up in the cortex. It's kind of the first thing to note. The process that takes place here is glomerular filtration. Alrighty, we can list the barriers for filtration. So remember, we've got the endothelial cell. Okay, and I'm going to put in brackets here that they say fenestrated capillary. We've got the basement membrane. And then we've got the epithelial cell, which is the visceral layer of the Bowman's capsule. So the epithelial cell, and remember we call that cell a podocyte. Okay. All right, so that's the location, that's the process. This is the membrane that we're crossing here, which is this membrane here. We also want to remind ourselves about the Starling forces here. I'm not going to go into detail about the Starling forces, but the Starling forces, the Starling forces are acting in this area. Right, we talked about the 4 Starling forces, so you can go back and refresh yourself, remind yourselves of what those forces are and what direction they're in, and make sure you're able to calculate what the overall movement is if you're given values for those Starling forces. Okay, let's talk about the intrinsic regulation. So remember that intrinsic regulation takes place here. There were 3 mechanisms that we described, and this is basically what maintained GFR at 125 milliliters per minute, right? This is a rate, in the face of a mean arterial pressure between 80 and 180 millimeters of mercury. Okay, so this is basically what the intrinsic regulation is doing. It's keeping GFR at that constant rate in the face of a changing mean arterial pressure that is between 80 and 180. Okay. And just to describe those mechanisms, we had myogenic regulation, Okay, we had tubulo-glomerular feedback. And we had the mesangial cells. Okay. We also mentioned that there was extrinsic regulation. And this was involving the nervous system. And so here we saw that if mean arterial pressure was less than 80 or greater than 180, then GFR would either rise or fall accordingly, okay? I won't go into the mechanisms here, but we know this involved the sympathetic nervous system. Okay. All right, let's look at the proximal convoluted tubule and talk about what happens here. And I'm gonna kind of underline filtration because this is the main process that we've got here. So in the proximal convoluted tubule, first off, the structure here is also in the cortex. Some of the features that we see of these cells here is we see leaky tight junctions. We see microvilli, which form that brush border. Here we see unregulated reabsorption. This is really important. So unregulated reabsorption. Okay. And we see that solute reabsorption facilitates water reabsorption. So sodium is reabsorbed actively. Chloride is reabsorbed passively. And then because of the movement of those two solutes, water is reabsorbed by osmosis, which is passively, okay? Other important substances are also reabsorbed here. So we talked about the fact that nutritive substances, things that the body wants to reclaim, are absorbed here. Amino acids, 100% of the glucose is absorbed here. We see the reabsorption of bicarb that we just talked about as a means of balancing our acid-base state of the plasma, okay? And then let's talk about the capillaries that are associated here. We talked about the peritubular capillaries. So these are basically the network of capillaries that are intimately associated with the proximal tubule where that reabsorption is taking place too, okay? All right, so these are your peritubular capillaries. So these are the important takeaway points for the proximal tubule. Again, remember the key point here is that the reabsorption is unregulated. This is facilitated by leaky tight junctions, by the modification of these cells. And so there isn't any hormone that is acting here. This is not influenced by the nervous system in any way or by the solute concentration in the tubule. This is just happening unregulated and freely, okay? Let's move down to the loop of Henle. So in the loop of Henle, we know that we have the descending and ascending limb. So we have the descending. So the descending limb that is only permeable to water. The ascending limb is permeable to sodium, potassium, and chloride. So our solutes. And this is what establishes the medullary osmotic gradient. Okay. And so the medullary osmotic gradient we talked about, which is established by the counter current multiplier. And the counter current multiplier, don't let this terminology confuse you. Remember, this is nothing but the fact that the ascending limb is permeable to solutes only, and the descending limb is permeable to water only. Okay, so here we can only move solutes, and here we can only move water. And this is what the counter current multiplier means. When this happens over and over and over again, then we establish this osmotic gradient, okay? We also pointed out here that there's the vasa recta that is intimately involved here. And so the vasa recta is the network of capillaries that is associated with the loop of Henle. Okay, remember the vasa recta has an opposite orientation to the loop of Henle, meaning that the descending limb of the vasa recta is the ascending limb of the loop of Henle and vice versa, okay? And then we spoke about the role of urea here. So urea is helping to maintain that medullary osmotic gradient. Okay, the fact that the movement of urea facilitates the movement of water, and the fact that the urea channels are only on one side of the loop of Henle is what helps to prevent the dissipation or the watering down of the medullary osmotic gradient. Okay, let me just take a peek and make sure there aren't any questions. Okay. I hope you can read this writing. It's a bit small, but it should be clear. Okay, now the last parts of the tubule, we kind of combine and talk about them together. This is the distal tubule and the collecting duct. And the takeaway points for this is that, actually, we forgot to mention here, the location of the loop of Henle is down in the medulla. Okay, so you remember the cortex, and then this is down in the medulla. Here in the collecting duct and the distal tubule, we have regulated reabsorption. Okay, and here we see the absorption of sodium, potassium, chloride, although we did not speak about chloride, and then water. And all of this is regulated. And again, just to kind of reiterate what I mean by regulated, this means that depending on our state, depending on the osmolarity of our plasma, depending on hormones, depending on our mean arterial pressure, these can be changed to either reabsorb more sodium, potassium, chloride, water, or secrete more sodium, potassium, chloride, water, okay? The secretion that we see here is potassium and hydrogen ions, right? So potassium as a part of regulating our solute constitution here, and then hydrogen ions as a part of regulating our acid base. We also want to point out the different features that we see here. So we see tight junctions. Meaning that they do not allow for any paracellular transport, the way that we see with the leaky tight junctions of the proximal tubule. Let's talk about the hormones, right? So because this area is regulated, it's obviously influenced by hormones. Let's talk about the various hormones and what their effects are here. Okay, let's not try to jumble this up. Okay. So the hormones here are ADH, or vasopressin, antidiuretic hormone, which facilitates water reabsorption. So we have ADH. And this facilitates water reabsorption by inserting aquaporin 2 channels. Okay. Let's put the 2 above here. Then we have aldosterone. Okay, and aldosterone is going to reabsorb more sodium or secrete more potassium by influencing the sodium-potassium pump. So one, it's going to be the sodium-potassium pump on the basolateral surface. And two is going to be the leak channels on the apical surface. Okay, and so if it inserted more of these things, we're going to reabsorb more sodium or secrete more potassium. If it inserted less of these things, we'll do the opposite. Okay, so both of these mechanisms will reabsorb sodium or secrete potassium. And I apologize, the space is getting quite small, but hopefully you can see that. So aldosterone via inserting sodium-potassium pumps, via inserting leak channels for both sodium and potassium on the apical surface, will either reabsorb more sodium or secrete more potassium. Okay, and then finally we have ANP, and ANP is doing the opposite. Atrial natriuretic peptide. ANP is going to decrease sodium reabsorption by blocking the leak channels on the apical membrane. So decrease sodium reabsorption, okay? And it does so by blocking the leak channels on the apical surface. Now, I want to point out here something that we spoke about as we talked about balance. So this idea of solute and water balance. So although ADH has a main role of influencing water reabsorption, understand that as a secondary role, it's also going to influence sodium reabsorption. And the reverse is true. Although aldosterone has a main role of influencing sodium, right, so of solute reabsorption, it has a secondary role of reabsorbing water, right? So it has a kind of hand-in-hand role with ADH. And then ANP will also have a secondary role of influencing water reabsorption, as well as influencing sodium reabsorption. Okay. All right, let me see if there are any questions here. I don't see any questions. I don't think there's anything else I forgot to point out. Oh, let's add here the names of the cells. So remember in the distal tubule and the collecting duct, the names of the cells here were the principal cell. So these are the cells that these hormones are acting on. And then we also have intercalated cells. Okay. And then also remember that this aldosterone, I'll put it here in red, is acting on behalf of the RAS system. So I won't go into detail about the RAS system, but remember, the renin is what creates angiotensin, is what eventually creates aldosterone. So the stimuli for renin are going to be the same stimuli for aldosterone. And in terms of a general picture, the stimuli for renin are going to be anything that decreases mean arterial pressure. So hemorrhage, dehydration, shock. And then the stimuli for ANP are going to be anything that increases mean arterial pressure. So think about chronic hypertension or chronic sustained high blood pressure. Okay. All right. So I think that is it in the way of